Okay, we have been discussing different complement pathways, and by now you guys would know it very well that the principal complement pathway is the alternate pathway that we have discussed in detail. Okay, but complements are not only activated by that particular pathway, although alternate pathway can be activated by different viruses, different bacteria, or parasites, or different immunoglobulins like immunoglobulin A, or snake venom, for instance. All these different things can activate the alternate pathway. But how are the other pathways activated, and why there is a need to have another pathway? Okay, so these are alternate options for the alternate pathway that we are discussing right now. There are a number of complement proteins that are present inside our bodies, and we have discussed them, that more than 30 proteins synthesized in the liver are there in the body. But they are not active, and there needs activation. And they are a very important weapon of the innate immune system. Okay? And the most abundant of all the complements is C3. As we were discussing about C3, that is the most important and the most abundant complement in our body. Okay, 1.2 milligram per milliliter of your body fluid is just C3, or the complement factor 3. And that is the main factor. All these pathways ultimately have to lead to the fixation of C3b on the surface of the pathogens, because that is what the immune system would recognize. If C3b is fixed to the surface of a pathogen, it will be readily and very actively phagocytosed or opsonized. We call it opsonization. Okay, and that is a way of killing the pathogens. Okay, so the lectin pathway, this pathway is actually activated without antibody, like the alternate pathway. Although I told you that in alternate pathway can also be activated by immunoglobulin A. Okay, but there are so many other things that can activate the alternate pathway. So we just skip that part of the immunoglobulin A, which is quite uncommon. Okay, so that's why we just say that it is activated without antibody, like the alternate pathway. And if you look at the events that are happening here in the lectin pathway, these events are more like the classical pathway. Okay, why these are common to the classical and the lectin pathway, you would learn in this lecture. Okay, for instance, the complement factor C4 is utilized both in the lectin pathway and in the classical pathway. The series of events that are going to take place in the lectin pathway and the classical pathway are more or less the same, but activation of the complements factors, that is different. The initial step, or the first step, is different, and after that the rest of the steps, they are the same. They lead to the same consequences. Complements would be activated. Ultimately, all these complements would lead to the fixation of C3b on the surface of the pathogens, and there would be more fixation of the C3b molecules, because C3b, can very easily do that. It associates itself with factor B and take a big part of the factor B and start, cleaving more C3 into C3a and B and fixing all those on the surface of the pathogen. Okay, because for C3b, as I told you, there are receptors on the immune cells, and the immune cells are going to phagocytose this particular pathogen, or take this pathogen inside and degrade it. Okay, so in the lectin pathway, this mannose binding lectin, this is normally present in your body. Okay, and this actually binds to sugar residues like mannose found on microbial surface polysaccharides such as lipopolysaccharide. Okay, these are sugars present on the surface of certain bacterial species, for instance, but not all of the bacterial species. Okay, and the lectins which are present inside your body have a tendency to bind to these sugars. Okay, and on the surface, these polysaccharides actually are present on the surface of these pathogens like Salmonella typhimurium, Listeria, Neisseria, Candida, albicans, etc. So what happens after the sugar on the surface of the pathogen is bound to lectin inside your system? Okay, these bacteria have entered your body. Okay, these have got these sugars, and these sugars have perfect partners inside your body in the form of lectin. So what would the lectin do? The lectin would just go and bind to its partner, which is the sugar present on the surface of the pathogen, on the surface of this particular bacterium. Okay, so this is now a perfect assembly for other factors to be activated. For instance, this is MASP1 and this is MASP2. These are mannose associated serine proteases. These are enzymes which have serine in their active pockets, and they can cleave other factors or other proteins. That's why we call them serine proteases. Okay, so what happens here, that MASP1 or the mannose associated serine protease 1, when finds such an assembly of the lectin with mannose on the surface of the pathogen, this is a perfect invitation for MASP1 to come here and bind to this assembly. Okay, it recognizes this assembly of the sugar and the lectin and comes here and binds to that particular assembly. That is the mode of activation of MASP1 or the first serine protease. Now this binding, another factor is there, which is MASP2. It not only it is not activated yet, but it finds a perfect binding site on the same assembly. Okay, it comes here and binds with the same assembly of lectin and sugar. So what happens next is that MASP1, the first partner, doesn't like this partner to remain intact as MASP1 is there in its active form. So it would readily go for cleaving of the MASP2. And when MASP2 is cleaved, this cleavage of factors or proteins actually activates the protein. This cleavage exposes certain bonds inside, which are hidden inside, the peptides. Okay, so that's why all the enzymes, their way of activation would be just like this. Okay, some factor would be there inside your body or inside your tissues, which would cleave a particular peptide and expose the bondings that are hidden there. That is a conformational change that is happening here. Okay, and those bonds then, when are activated, or those sites when activated, they would just go for cleavage of other factors. Okay, but when the bonds of the MASP2 are hidden here, it cannot activate further complements in this pathway. Okay, so that's why MASP1, once bound here, was activated upon binding, but MASP2 was needed activation, and MASP1 just took a piece of MASP2, divided it into two pieces, and one of the piece which remained bound to this assembly of the lectin and the sugar now is an active serine protease. So what the active serine protease would do, we would discuss here in these images. But first, this is not the only job that these serine proteases are doing inside our bodies. You should also remember this, that MSP1 can also cleave coagulation pathway proteins such as PAR4 or fibrinogen or factor VIII, which leads to high clot and fibrin generation. Okay, so they are not only involved here in activation of the lectin pathway or activation of different complements in the lectin pathway, they are also involved in the coagulation pathway, and clotting which happens here when somebody is injured. You see the different clots formation, which include these different factors like fibrinogen. Fibrinogen is an inactive form, okay? But fibrin is generated out of the fibrinogen with the help of MASP1, okay? Similarly, other factors. So these other factors are also cleaved by MASP1. The cleavage brings about activation of these complements or these factors, and MASP1 is doing these jobs in other pathways as well. Okay, so now how the activated MASP2 is going to lead the things. Okay, activated MASP2 cleaves C4 to C4A and C4B. Some C4B binds covalently to the microbial surface. Okay, this is the assembly that you see here. In this assembly, MASP1 activated MASP2, and now here MASP2 is activated. Okay, now what is this activated MASP2 or serine protease doing? This, when this kind of assembly, when MASP2 is activated here, this is a perfect invitation for this complement number 4, to come here and bind to this assembly. There is a binding site for that. These are all energy dynamics. As the energy dynamics of molecules change, the bonds are made or the bonds are broken. Okay? So this is binding actually is also because of that attraction, that you have been studying in your chemistry course, for instance, making and breaking of bonds, which is dependent upon electrons that are leaving or joining some particular molecules. And you know about that. You are very well with chemistry, I think. So this C4 complement comes here and bind to this assembly, and this binding is not very, fruitful for him or her. And what happens that the MASP2 would cleave this into two parts, a smaller part and a bigger part. And the bigger part of the C4 complement just falls down on the surface of this pathogen because this is heavy. Okay? So it's not only heavy, the bonds in C4 are exposed just as the bonds in C3 were exposed when C3 was cleaved into C3b and C3a. So just for ease, you can just say that what molecule would attach with the pathogen surface, the molecule that is bigger in size also always will fall down. Okay, so it is falling down and just fixing to the surface of the pathogen. This is C4b, the bigger part. Okay, so of course it is making covalent linkage just like the C3b did in the alternate pathway. Now this assembly is not only deceiving this complement number C4 to come and bind to its perfect place, it's inviting him to come here in the place is now ready for you, and you should have your own position. So this is also inviting other factors that you also have a space. You can also come and bind to your perfect bonding site. So C2 is the second complement, which is also deceived in a similar way. It comes here and binds to the perfect place and probably thinking that it would take some rest here. Okay, but this complement has invited him in order to cleave it into two parts, a smaller part and a bigger part. Here the smaller part is known as C2b and the bigger part is known as C2a. So as this is a bigger part, now this bigger part will fall down on the surface of the pathogen, and when it falls down just over the head of C4b. So it means that C2a and C4b, they are complementary. They can bind with each other. There must be a binding site on C4b for binding with this bigger fragment of the C2 complement. Okay. Now this C2a and C4b, this combination actually is the classical C3 convertase. Okay. This is the C3 convertase in both the lectin pathway and the classical pathway. So this C3 convertase would do what now? Okay. This C3 convertase, as the name indicates, would recruit C3 molecules and hydrolyze or cleave the C3 molecules into two parts, just like it is doing here. You see here, now this one is free, it did its job, okay? Now here is C4b, which is bound to C2a, and they have invited another partner, which is C3, and cleaved another partner, C3e, into A and B parts. And the B parts is fixed to the surface of the pathogen, and that was the ultimate goal. The ultimate objective of all this exercise was to fix C3b to the surface of this particular pathogen. Okay, so the story started with C4 and it involved C2 then, and then finally it ended with C3 and the fixation of C3b on the surface of the pathogen. So the consequences of C3b fixations are the same for the pathogen. This is marking, this is labeling of the pathogen. This is going to lead to the destruction of the pathogen or killing of the pathogen, because this molecule has got receptors on immune cells, and immune cells can recognize everything that is labeled with C3b. It can opsonize and it can destroy that particular structure, because C3b fixation can not only lead to this consequence, it can also lead to membrane attack complex, which is formed on the surface of cells, and in this way you can say that the pathogens are destroyed.